Okay, Pauline, what'd you put for number one? Comparing uh, cytokinesis, telophase.
Animal cells create a cleavage furrow while plant cells form a cell plate.
Ooh, yeah, so animals use a cleavage furrow, plants use a cell plate. So, eh. So, basically, a piece of cytoskeleton just pinches the cell in half and creates that cleavage furrow, actin ring, and it can just break it, just slowly keep tightening and breaks or separates the two cells. And we see in plants use a cell plate where they're just building a brand new cell wall in the middle. And the reason, and what's the reason, anybody think, why can animals just pinch it into two pieces and plants can't? Like, why can't a plant cell just pinch it and have that work? Like, what do you see? What's the main problem with doing that with a plant cell? What's surrounding the plant cell? What's that, America? Cell wall, right? Is cell wall pretty rigid? Strong? Yeah, you just can't pinch that in half. You're going to have to build a whole new wall. Whereas the cell membrane is fluid, the phospholipids can move around, and the proteins, and it can be pinched and kind of squeezed and separated like that. Okay? So, today you guys are going to be looking at plant cells doing their parts, doing their phases of mitosis. Okay, which is all going to be focused on looking at the DNA/chromosomes inside the cell. That's the big focus of mitosis, looking at what's happening with the chromosomes. So in that graphic at the bottom, what do you notice about that, Emily? Anything pop out? It's like, what similarities or differences you see there? Anything? I don't even know what I'm asking this question. I'm just kind of putting it out there. So what do you see? You want help? Not seeing anything? No, nothing coming to mind? On the left, there's what? On the left, there's what? Strands? How many strands do you see? 2, right? 2 strands. Because, as you know, genetically, DNA-wise, what percent comes from your mother? 50. What percent comes from your father? 50. And that's because you each get 1 chromosome from each, so they add up to half and half. So that shows, the colors are indicating the chromosomes that you receive from the mother and father. Okay? So you have 1 chromosome, and what do you have on the right? What happened to those chromosomes? What do you see? Or what do you read? There's a word up there. Kind of describes what happened. You want some help? Okay. Let's go to Miguel. What do you notice?
the single strand basically divided itself and then they joined together.
Ooh, so we have the single strand here. Over here, is it a single strand? No, it's now a double strand. Because what did that strand do? What does replicate mean? If we replicate you, Miguel, what have we done?
Another U.
Made another U, right? So we did that. That was done with the chromosome. So you have an exact copy. You have a replicated chromosome. Okay? This is what happens in mitosis because you need to have equal amount of genetic material going to each new cell. So you double the DNA, you replicate it. Okay? So we see that each chromosome is replicated, copied, doubled. Yeah, each chromosome. So I can slow. Now it has 2 copies. Now this has 2 copies. 1 copy. Now we have 2 copies. So 1 can go to each side. So everything stays equal. When you think of chromosomes, like when I think of chromosomes, usually that kind of X formation comes to mind. The copied formation, when in reality the normal status for a cell is for it to be in this formation, just 1 chromosome. Okay? Probably because, when we look at these, we're always looking at them, or not always, but sometimes, many times looking at it in mitosis. Okay? So, copied chromosomes get separated. You'll see sister chromatids. That word sister chromatids you'll have to know. So those sister chromatids are copies of each other. They're the exact same. Okay? So whether you get the left side or the right side, everything will be the same. Okay? All right. Well, I want you to look up at the board. And these are simplified diagrams of what we're going to be looking at today under the microscope. So these are all different cells in different phases of their cycle. This right here is interphase, where you just have the DNA spread out. It's all, it's not condensed. It just kind of looks like, I don't know, just a bunch of specks under the microscope, a bunch of colored specks. And that's overwhelmingly what you'll be seeing. Now, once you get into the phase of mitosis, that DNA is going to start to form these little worm-like structures, which are the chromosomes. And they'll all be in that kind of circle starting prophase. Continuing, you get into metaphase. These spindle fibers will start organizing the chromosomes so they're right in the middle. And this will kind of look like a bunch of like little worms kind of stuck in the middle in a bunch. Then you'll see they start separating themselves. These are going to go to the top, these are going to go to the bottom. And then that brand new cell wall is created. And the chromosomes start to unwind, they go back to being normal DNA, and the nucleus then reforms. So, those are diagrams. Do things look different in reality? Yes, they do. So this is what they look like in real life. So, just kind of the colors all spread out and even. That's interphase. Once you see some clumping like this form, That's your tip off, you're going into prophase. Now, can it be hard to tell between interphase and prophase? Yes, there's an exact line in between. It's tough to have sometimes. You're not going to see it super clearly all the time. So you're just going to do the best you can when you identify these things. All right? So prophase, prophase. All right, now we're getting something different. These are the chromosomes, kind of looking like worms. They're kind of bunched in the middle. That's metaphase. Another example of metaphase right here. All the worms bunched in the middle. Now, they're starting to separate. It's moving into anaphase. This is a later part of anaphase. They're almost on either side of the cell. But there's no cell wall yet. So we call that anaphase. But that can be tricky to distinguish between anaphase, the late anaphase, and then telophase here. Telophase, you see how that cell is a little bit smaller? Like that is not big enough to be a full cell. So what this probably is telophase. It's just getting divided. Both of these cells are smaller than the normal cell size. You still see some worm-like structures. That's tip off, you're in telophase. All right. Any questions? So let's go. The pen response is a little bit slow, but can somebody follow along with me on this while I go up at the screen? Anybody willing just to label this? Oh no. Uh-oh. I guess we can't do that. So I gotta get this on the screen here. Charger. Okay. All right. Well, let me just do this way. So let's just start at the very top left and just start labeling these. So what do you guys think this one is? Can you see the DNA as it clumped up in the middle? Oops. It looks like we got some worms in there. It's kind of in the middle. I would say that's prophase. So I'm going to put a P right there. Oh, it's a little faster. What about this one? Definitely, it's a phase of mitosis. See those big worms. Where are the worms located? What do you think, Jacqueline? What would you choose for this one? Prophase, metaphase, anaphase, or telophase? What do you think? Yeah, I would call that metaphase. They're bunched in the middle. You can see them. Metaphase. The next one over. You can see it pretty well. It doesn't really look, I can't see a lot of clumping. It looks like just regular interphase. The next one over. Actually, the next three look the same, don't they? Those all look like interphase. Remember, interphase is going to be by far the most common one. Oh, that looks like anaphase, doesn't it? You can definitely see them pulling apart. And then, the next. I don't see any clumps. They're all separate.
There's 2 clumps.
Well, you see, in their normal interphase, you will sometimes see 2 clumps from the nucleoli. So those there, that looks like regular interphase. But it can be tricky. So you're just going to do the best you can. Now that one, it looks different, doesn't it? It looks like you can see the worms there. So this would be prophase. The next one over, that looks like interphase. Let's go down to the next one. There we go. This looks like interphase. Ooh. I can see a whole bunch of squiggles in here. It's pretty big. That's probably prophase. Oh, this one here? Those are kind of on the smaller side. And I see I can see some definition here. So I think this is telophase cytokinesis. I'm gonna go ahead and roll the dice, call it telophase. This looks like interphase. Ooh, that's telophase. We can see that, right? We can see it, but just barely forming. And then a whole bunch of interphase. Let's do one more. Can we see these first few? I'm just going to skip that. That one looks like interphase. Can't really see that too well. Interphase. Ooh. Prophase. That's a cool one. That's a cool anaphase. Well, they're pretty small, but they look, I don't see any condensed. I'm going to call these these are both interphase. Ooh. This looks like anaphase, late anaphase. They're on each side, but I don't see a cell wall yet. Interphase, 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 interphase. Okay? So that's what you're going to do under the microscope. Just go through, and I want you to try to get 100. Okay? Count 100. Okay? And what you're going to be doing is you're going to be making tally marks. Interphase.
Right here?
Yeah. Interphase, prophase, metaphase. Hold it. Anaphase, telophase. Okay. So how many interphases do we count here?
Interphase.
1, 2, 3, 4, 5, 6, 7, 8, 9,
22, 23, 22.
I count 24. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25.
Yeah, 25.
So this would be
25, 10.
okay, and then prophase. How many prophase? 4?
Yeah. And then
Metaphase.
1.
I think there's 1, right? Anaphase.
1, 2, 3.
1, 2, 3.
1, 2, 3.
We have 2 telophase.
2 to
All right, and how 25, 24, no 30, so 35 total. Okay, that's our total. So to get the percentages, like you see that second column over, you're just going to take, for example, interphase 25 divided by 35. What's 25? Got a calculator? Get your phone out, everybody. Everybody get your phone.
25 divided by
Yeah.
0.7.
Is it 0.70?
1.
0.71?
Yeah.
All right, so 0.71 and multiply by 100, that's 71%.
Times.
Okay, so 71% are in interphase. And then the next column over, you want to get how many minutes that represents for the cell cycle. So you're
0.71.
going to take 0.71 times 1,440.
This is wrong. Is this right, mister?
Should be like a big number. 1,000?
Or did I do something?
Yeah?
Yeah.
So 1,000 minutes,
It doesn't mean it. It doesn't mean 22.
1,022 minutes. And that makes sense, right? Because that's the majority of 1440.
It was easy.
Okay. let's see. So we'll try for 100, but we'll see. 75 might be good. Okay? Which you'll just, whatever the total is what you divide by right here. Okay? So I do want you paired up, okay, on the microscopes. And I would suggest getting a picture of the mic. Once you have a good thing, get a picture, and that's going to be easy for you to do your counts and your tallies. You might have to move it around a little bit. Right now, they should be. Well, actually, no. Before you get there, go ahead. They were set up. I'm not sure if they're still all focused under high power. You will be under high power the whole time. You might have to move it around a little bit to see different parts of the onion cell. Okay? All right, let's go. 2 people per microscope. Or you could go solo if you want.